The first and arguably most important derived index is the mean corpuscular volume, or MCV. The MCV measures the average size of a single red blood cell. This is the principal index we use to classify anemias morphologically, that is, by the shape and size of the red cells. If the MCV is low, the cells are smaller than normal. We call this microcytic anemia, which points us toward causes like iron deficiency or thalassemia. If the MCV is normal, the cells are normal-sized, normocytic anemia, which has its own differential. And if the MCV is high, the cells are larger than normal, macrocytic anemia, which raises the question of B12 or folate deficiency, among other causes. MCV is your first morphological sorting tool when you approach any anemia. The second index is the mean corpuscular hemoglobin, or MCH. The MCH calculates the average absolute amount of hemoglobin contained within a single red blood cell. Think of it as asking, how much hemoglobin, in total, does each individual cell carry? It gives us a quantitative sense of hemoglobin content per cell in absolute terms. The third index is the mean corpuscular hemoglobin concentration, or MCHC. The NCHC refines this question further by calculating the average concentration of hemoglobin inside a single red blood cell, that is, the amount of hemoglobin relative to the cell's actual volume. Why is this useful? Because a cell can have a normal absolute amount of hemoglobin but still be abnormally concentrated or dilute depending on its size. The MCHC is particularly important in diagnosing conditions like hereditary spherocytosis, where the red cells are abnormally small and spherical, causing hemoglobin to be packed more densely than normal, producing an elevated MCHC. That elevated concentration is a diagnostic clue you do not want to miss. The fourth index is the red cell distribution width, or RDW. The RDW quantifies the degree of variation in the size of red blood cells within a blood sample. In a healthy individual, red cells are fairly uniform in size. When pathology is present, that uniformity breaks down. You get a heterogeneous population of red cells of varying sizes, a condition we call anicytosis. The RDW captures this variation numerically. An elevated RDW is an early and sensitive marker of nutritional deficiencies. It rises before other parameters become obviously abnormal. In iron deficiency anemia, for example, the RDW often becomes elevated early in the disease course as the marrow begins producing smaller, iron-poor cells alongside the residual normal cells. It is also a useful signal in mixed deficiency states, for instance, combined iron and B12 deficiency, where you may have both microcytes and macrocytes in the same sample, producing a wide distribution width, even when the MCV looks deceptively normal.